What I want to do is just take a brief step back just to review that excitation-contraction coupling in the smooth muscle. As you guys noted last time, in that there's some differences in the smooth muscle versus a skeletal muscle. I mean, you could even say simply, well, they're built differently, so they're going to operate differently. Even though they're both muscles, they're going to get excited differently and they're going to contract differently. So the first thing is we learned that smooth muscles not only have the nervous system controlling them, but some smooth muscle cells have an intrinsic source of control from the pacemaker cells. And in this figure 1235 is illustrating looking inside of a contractile cell, a muscle cell that contracts. So we're saying, well, no matter where the source of excitation comes from, whether it's from an autonomic nerve exciting it or an intrinsic or a pacemaker exciting it, the contractile cell, after it receives that source of stimulus, has to let calcium into the cytoplasm. And that was the first big difference, is where that calcium originates. In their smooth muscle contractile cells, our calcium actually comes from where? From the extracellular, right? So we've got these voltage-gated calcium channels across our smooth muscle contractile cells, and when there's a stimulus, they open up, calcium moves down its gradient into the cell. That then triggers a secondary flux of calcium from where? The smooth ER. Okay? You can call it the sarcoplasmic reticulum, because that's the modified smooth ER. So we're just adding to our source of calcium. Then we got to a good fun second messaging pathway. And what does calcium do once it's in the cytosol? Very good. Good job. It binds to something called calmodulin. As the name kind of gives itself away, it's going to regulate something. It binds calcium. So calmodulin is an intracellular protein in our contractile smooth muscle cells. They bind together, and they're shown complex to here in this little purple thing. Does calmodulin look like that? No. Okay, we're just keeping it simple, right? Calmodulin now acts as a messenger, and what can it bind to? When it's bound to calcium, it binds to something called myosin kinase. What is myosin kinase? It's an enzyme. Yes. It phosphorylates myosin by substrate level phosphorylation. Good. So myosin kinase tells us, ASE tells us it's an enzyme. Kinase tells us it's going to phosphorylate. Work backwards. It phosphorylates, allows for the phosphorylation of myosin. Again, as I tried to highlight yesterday, or excuse me, on Tuesday, we're going to follow the phase of excitation. Right? So when the myosin cross bridge is phosphorylated, we gave it a phosphate, but functionally, what are we giving it? Energy to contract. Very good. Okay, so it's going to contract, and we'll do the cross bridge cycling. The smooth muscle will maintain that contraction for however long. Till when? There's more ATP and even more calcium. Right? As soon as the calcium is gone, then the muscle will relax. But as long as calcium is in the cytosol, the muscle will contract. So smooth muscle will maintain contractions a bit longer than, say, when contracted to skeletal muscle. So it may maintain for several seconds or longer, if you will, when compared to a skeletal muscle.
Is myosin kinase, you call it that green thing?
Uh-huh.
That's the same as myosin light chain kinase.
Yes, the light chain is just getting very technical about it. Because what it does, it puts a phosphate on the chain part instead of the head part.
Okay.
The tail instead of the head. So the myosin is different in smooth muscle than it is in skeletal muscle. But it's still a myosin protein contractile protein. There's something just flat out wrong in your textbook. And we didn't address it, so Before I tell you what's wrong about it, is does calcium bind? Okay, does calcium bind to the thin filament in smooth muscle? No, it binds to calmodulin. So calcium, yes, regulates each skeletal muscle and smooth muscle contractions, but differently. Okay. So in the skeletal muscle, calcium binds to what? Troponin, right? Okay. In the smooth muscle, it binds to calmodulin. Okay. So completely different, right? Right, protein. So what that tells us, if calcium doesn't need to bind to troponin, the smooth muscle, does it need to have troponin? No. Okay. So your book says smooth muscle cells have troponin, but no tropomyosin. It's completely backwards. They have tropomyosin, but they don't have troponin. Okay, so. Here's the thing. One, they're just wrong, and then maybe they just found bad data. I'm going to tell you one thing, is the data, we're still really just learning about smooth muscle contraction. That's when you go, if you were to go on your own and look about smooth muscle contraction, the number of data or the journal articles just plummet when compared to skeletal muscle, right, physiology studies. Because we're just really kind of putting it together. So they may have, when they put together the book, got just some bad data. I found some recent data from 2010 looking at the structure of our thin and thick filaments here, but and actually both are absent in this image as well. But there'll be more data to support. There's tropomyosin, not troponin. There's something very similar to troponin in the smooth muscle, but it's not troponin. Does that make sense? Okay, so there's another regulatory protein, but it's not actually troponin. And so there's just different mechanism for excitation. If we were to look at a tiny little contractile component of the smooth muscle, we'll see the thin filaments as shown here in the little brown spheres, and the thick filaments in the yellow. And the question was, well, what's the light chain bit? We phosphorylate on not the head, but on the chain itself. Does that make sense? Where the phosphate gets transferred in there. Right. So anyway, just myosin kinase helps put that there. Okay. I think that's the primary thing that I wanted to show you guys. When we look at a smooth muscle when it's in the relaxed versus the contracted state, we still have overlapping between the thin and thick filaments. But because the way that the thin and thick filaments are arranged, we can't see dark, light, right? So we can't see the striations, but the contractile machinery is still there. And so when a smooth muscle contracts, it shortens and goes, kind of use the word splay? Does that make sense? It splays out, it shortens and splays. It's not just completely short and broad, it like splays out. I don't know if someone know of a better word. So watch my hands, it goes short and splays out. It shortens and, can I do this? If I were a muscle, it would split. Does anybody know another word that I could use? Shorten, sometimes they say broaden. Layers, I like that. I like it of short layers, a little bit. Splays. Because your smooth muscle cells aren't connected to bones, right? So they're attached to something way on some other part of the body, so it kind of it shortens, broadens, and right depends on where. If you're looking at it and say the uterus versus the bladder versus the digestive system or something like that. Spreads, widens. Spreads and widens. Kinda. We turn to figure 1236. Okay, this should look familiar from your handout. Just take straight from there. Putting a visual comparison between our multi-unit and our single-unit cells. To review for you, we learned or studied last time that our multi-unit smooth muscle cells are strictly dependent on extrinsic control from the autonomic nervous system. They don't have any pacemaker cells to keep all these cells working together. By contrast, the single unit smooth muscle cells, yes, they're controlled by the autonomic nervous system, but they're also connected by what? Gap junctions. Okay. And so that there's going to be a pacemaker cell spontaneously depolarizing to excite the rest of the cells. And so on your handout, I just said pick one because these are all super generic looking cells, right? So you could have just picked this one or this whatever. Pick a cell, call it the pacemaker cell, and then it'll send action potentials to all the other contractile cells that it would be connected to. Does that make sense? That looks like that concludes our discussion for smooth muscle cells. So I want to field any questions that you may have about smooth muscles before we contrast them to cardiac muscle, which is the third thing in this. What questions do you have about smooth muscle contraction, control, structure, function?
Okay, so action potential isn't the same as far as skeletal versus
No. Did we look at those? In the pacemaker cell. Okay. In the pacemaker cell, the action potential happens due to an influx of calcium. So the difference will be that leakiness to calcium.
it's like as far as the picture though, like the picture is still like it has to reach threshold.
Yes.
Threshold the same number?
It will be probably slightly different. I don't know the actual values of threshold in a smooth muscle cell. This image is going with -50, so that might be a good threshold level for smooth muscle cell levels. But it's the same concept. Your cells maintain a rest. They can depolarize across it. They are up to it and then across it. Getting great. Yes. So let me say it this way. Smooth muscle cells will generate action potentials, yes. Is it identical exactly to skeletal muscles? No, the ions are a little bit different. But the concept is the same. Threshold is maintained, got to grade up to it, or depolarize up the threshold, and then cross it if you want to see an action potential conducted. Good question. Additional question? Clarification? Going once? Smooth muscles going once. Cardiac muscles, it is. We'll do a brief introduction to the cardiac system, and then we'll transition into chapter 13. I just want to very briefly compare and contrast your cardiac muscle from your skeletal and your smooth muscle cells. Skeletal muscles, we said they are striated. And within those striations, making up those striations, you have your thin and thick filaments. And if you recall, our thin filaments, yes, have actin, but they also have what? Troponin, and then also tropomyosin. They're arranged, the sarcomeres are arranged in the thin and thick filament, right, or the light and the dark bands, excuse me. And so your cardiac muscle also is striated. It also has troponin and tropomyosin, and it also has that banding, right, pattern, right, that striation that's implied. So that's a primary similarity of cardiac muscle to skeletal muscles, that striation. From then, you'll see a lot more similarities of cardiac muscle to smooth muscle. The first thing is cardiac tissue is like single unit smooth muscle because the heart has a pacemaker, a set of cells that spontaneously, regularly depolarize up the threshold, generate action potential. So your heart is like single unit smooth muscle with the expression of our pacemaker cells. So that tells us heart has a source of what type of control? Intrinsic. The heart is intrinsically controlled. It is also, I'm going to pop down to the bottom of the list, it's also extrinsically controlled by the autonomic nervous system. So our cardiac muscle is like our smooth muscle in that there's the intrinsic pacemakers. There's also the extrinsic control from the autonomic nervous system. It's dually innervated. Also like smooth muscle is that our source of calcium initially comes from where? You said it comes first from the extracellular fluid and then triggers a secondary release from the smooth ER, the sarcoplasmic reticulum. So it's like smooth muscle in that its first source of calcium comes from the extracellular fluid. And then secondarily from the sarcoplasmic reticulum. And another common thing between the cardiac muscle and our single unit smooth muscle is the expression of gap junctions so that our pacemaker cells can coordinate the activity of the contractile cells. Do you guys have additional comments or questions about this introduction? We'll conclude this presentation and we'll move into chapter 13.